Thank you all for joining our webinar today on accelerated phenotyping and biomarker structural elucidation at the National Phenome Centre. My name is Nancy Wright-Ross, and I will be your host. At the end of the presentation, we will have time for a brief question and answer session. We ask that you use your Q&A box during that time. If we're unable to get to your question, you can contact our speakers offline. I would now like to introduce our first presenter, Dr. Matthew Lewis. Matthew joined the Department of Surgery and Cancer at Imperial College London, known as ICL, in 2010 after successfully working to establish the Metabolomics program in the core research facility at Colorado State University. He was recruited to ICL to oversee mass spectrometry operations within the Division of Computational Systems Medicine, which have since grown to include the Clinical Phenotyping Center, and following the 2012 London Olympics, the MRC-NIHR National Phenome Centre, known as NPC. As the Chief Operations Officer for the NPC, Matthew was responsible for the development and execution of analytical strategies, as well as the Center's portfolio of research collaborations, overseeing them from design to delivery. His research focuses on the development and application of high-precision methods for the metabolomic phenotyping in clinical medicine and population health, as well as the development of metabolite assignment and identification resources and capabilities. At this time, I will pass the presentation over to our presenter, Dr. Lewis.
Well, that's fantastic. Thank you so much for the kind introduction, and thanks to everyone online for joining today to hear a bit about metabolic phenotyping at the National Phenome Centre and our ongoing collaborative work with Bruker. With that said, I'll get started, move on to the next slide here. So just a brief introduction to the human metabolic phenotype. I think this is generally well appreciated in the field now, but worth covering, that we as humans, as superorganisms really, have our own genetic blueprint, which establishes a part of our physiology. But also, as we go through life, we have a number of different environmental exposures that we're subjected to. For example, foods, drugs that we consume, toxins and pollutants in the environment, in the space that we occupy, aspects of lifestyle, whether or not you have activity in your life, you're sedentary in your job, or you endure quite a bit of stress. These two aspects really culminate in the human phenotype, which is sort of the expression of those genotypic traits and the influence of all those environmental factors. Our group really also focuses on the microbial component of this, because as I said, we're superorganisms. We're not just human. We have complex microbial colonization in our gut, on our skin, elsewhere. And the populations can change, the genetics can change as a consequence, and we can control those with antibiotics or other means. So it's actually quite a complex organism that we study, and I want to tell you a bit about how we're studying human metabolic phenotypes. These are also, it's important to note, not just snapshots anymore, right? We really look longitudinally at human metabolism in terms of progression through different diseases, recovery from disease, different treatment regimes, a response to treatment, patient age, things of that nature. Phenotypes really are dynamic. So we're often looking for fingerprints within the human metabolome for use either as diagnostics or prognostics or as monitoring tools for health, etc. And these are just some of the areas in which we have active collaborations within the National Phenome Centre. And this is only to illustrate the fact that the platform that we maintain and that we're currently developing in collaboration with Bruker is really one that's very applicable for a number of different disease types. And in fact, even outside of clinical medicine and epidemiology, it's very a wide application space can be used here. Of course, this is an analytical challenge, though, deconvolving the analytical complexity that exists within the human metabolome. This is stated nicely, of course, by the Wishart Group in the Human Metabolome database. All you have to do is go to that database, and you'll appreciate the rich complexity that we as analytical chemists really have the challenge of tackling, deconvolving, specifically measuring, doing so rapidly enough to satisfy the larger scale studies that are starting to come through the field. So in the past, we have worked hard on establishing an analytical platform. It uses both NMR and LC-MS technologies. We specifically use LC-MS because we're usually working with human biofluids, liquid biopsies, et cetera. And our specialty here at the National Phenome Centre has been a very high-quality data generation on both platforms. And that manifests in different ways. I won't have time to talk about all of those today, but we have a few publications out there which explicitly detail the platforms and how we think about doing large block sort of continuous analysis to a high standard of quality with high precision data. Okay, together these tools and methods that we've developed culminate in the measurement of tens of thousands of chemical features, which covers a broad metabolic space, which allows us to assess fingerprints of, for example, human disease or human health, or for use in patient stratification. Will a patient respond well to a treatment, et cetera? So we're using this broad profiling technology in order to derive very specific and sensitive panels of biomarkers which inform us about patient health status. Okay, so what that means is that we have, as I said, an analytical platform. I said it's based on NMR and UHPLC-MS. Into that, we're typically putting urine, serum, and plasma. That's what exists in the clinic. That's what's most easily obtained from the clinic. It's what exists in biobanks and epidemiologists, freezers. So that's what we've done quite a bit of work with. As we continue to sort of adapt our platform for use in other more interesting study, we're now taking in cell culture and tissues, hard and soft tissues, as well as other matrices, human matrices like feces, saliva, et cetera. So this is part of our R&D platform, but really, I think more interesting for this talk is how we're transforming an analytical platform, one that I think is now commonly accepted within the field of metabolomics, into something that's a bit more meaningful in terms of being a well-characterized platform for measuring chemistry and measuring metabolism. So in order to do that, today I'm going to speak about three aspects of our collaboration with Bruker. One of them is relating to the use of NMR in terms of spectral deconvolution of the quantitative profiles that we're deriving from routine sample measurement. The second is accelerating de novo structure elucidation, something that our group has invested a lot of time into. And third is biofluid mapping. It's sort of an extension of the second. So I think we can take for granted that as samples come through our lab, we are analyzing them by standardized, now routine published assays that have been developed in collaboration with Bruker, and we obtain rich, informative profiles of the chemistry contained within urine, blood, and other sample types. But really quite interesting in terms of transforming our platform from an analytical one to a metabolism measuring platform is this new ability to deconvolve those profiles. And by that I mean looking specifically at signals which relate to quantitative measurements. So for example, using statistical deconvolution, we were able to look at broad signals in the lipoprotein region of these profiles, and we're able to deconvolve over 100 specific, distinct lipoprotein species measurements, and do so in a effectively a targeted and quantitative manner, providing reports that are similar to clinical in vitro diagnostic type feedback. So this has been really transformative for our collaborations, because not only are we providing ppm shift values and things that are typically associated with metabolomics data, spreadsheets of analytical measurements, but we're also able to provide things that look a lot like clinical chemistry with named components on it, which is very, very useful. Similarly, in collaboration with Bruker, of course, there's an increasing number of small molecules that are able to be derived both from urine and from blood samples and provided in this sort of clinical-esque quantitative manner. So this is a very important cornerstone of our transformation from pure analytics to understanding biochemistry. We're augmenting that both in NMR and in LCMS by the building of databases. Okay, I think this is an important thing when you have an analytical platform to characterize it to the greatest extent possible by pre-annotating as much of the signal content as possible. So this is something our group's been very active in, as well as the generation of spectral matching methods to ensure that we're able to connect reference data obtained from reference chemicals to data generated either by NMR or by LCMS, and thus we're able to put names on hundreds, hopefully soon thousands of chemical measurements and pass those along to our collaborators, even before we know whether or not they're statistically significant in any specific investigation of the data. Okay? But sometimes that approach fails. I think it's still true that our breadth of measurement in untargeted profiling exceeds our knowledge of what chemistry exists within the sample, which means that molecular profiling is still a high-value approach to doing investigation of complex human biofluids. Okay, so in the cases where our databases fall short, whether they're open source or in-house, we have to turn to de novo structure elucidation of unknown biomarkers. And this is an area, again, where we've been very active. In fact, we're expecting for a publication to be accepted very soon on this. So hopefully in the coming month, I would be looking for that. But just to describe to you kind of briefly how we've tried to accelerate this process, we've been able to take biofluid samples from studies or even just stock pools, right? Because often we're not looking specifically at silver bullet biomarkers that have complete presence and absence in clinical populations. Often we're looking at biomarkers that are present always in the population, but modulated with respect to different disease states. So we're pulling from one source or another biofluids and repeatedly fractionating them using no real special kit. We don't have a special investment in prep scale fractionation here. Instead, we use our analytical scale instrumentation. We just run it repeatedly in order to amass enough of the purified chemical in order to go on and do advanced mass spectrometry and in some cases NMR. So here we're actually working with Bruker to use their MRMS technology to take purified unknown biomarkers and perform some specific chemical formula elucidation utilizing the fine isotopic structure that exists and the ultra high resolution that the MRMS system is capable of generating. So this is actually an absolutely fantastic tool for clearly and confidently deriving a molecular formula, not just for a molecular ion as shown here, but through fragmentation experiments, we can do the same for all fragments that exist for a specific target molecule. As a consequence, it is really quite simple and often automated now using Bruker software to actually assign chemical formula to complete unknowns using this technology. In some cases, we're able to use their software. In this case, MetFrag, it's a nice sort of open source tool that exists on the web. They've built it into their software, and using that alone, we're able to make assignments confidently for quite complex molecules that we've been able to validate after the fact, and an example is shown here. Sometimes where that it doesn't work just using MS by itself, we have to use 1- and 2-dimensional nuclear magnetic resonance again, and in doing so, we're often able to elucidate novel markers, in this case, a novel marker of a kidney stone disease, indole 3-acetic acid glucuronide. And these are examples that will be in that forthcoming publication. So this is how we add value to our platform. It is a bit of a traditional approach, but I'll show you next kind of how we've worked to automate that and do as much of the work up front as possible. So we call this adaptation of the approach biofluid mapping, okay? We take a large biofluid pool, we do larger scale fractionation, in this case in multiple dimensions. We then bank the fractions that are made from the urine or from the plasma or serum separation so that we have material to go back to. And then we utilize these same tools, the MRMS, the NMR, also our routine UHPLC-MS assays, as well as our routine 1D profiling assays, which are being applied to all samples being measured within the Phenome Centre. And in doing so, we're able to start to translate between technologies. So, for example, a biomarker found in an LC-MS assay, we can look in our fraction bank and we can say that biomarker appears in this case, for example, fraction 87. Can we look in the NMR signature and see a signal that relates to potentially what we think from the mass spectrometry derivation of molecular formula, what we think that signal may be, okay? We can take this further and sort of use more data generation capabilities existing within the MRMS and the NMR platforms to, again, as I said, generate high confidence molecular formula assignment and also molecular structure assignment by 2D NMR. And we're able then to sort of translate among all of these technologies in a Rosetta Stone-like fashion, drawing the best of each technology and what we know, for example, the formula from MRMS, and being able to apply that to a biomarker that's observed, for example, in an NMR study. So this is a nice way of banking all of the information about any given biofluid in advance and reducing the amount of work that needs to be done when a specific biomarker arises. So an example of this, we take an unknown feature of interest from one of our routine profiling assays, in this case using reversed phase LC chemistry paired with positive mode ionization. The unknown in this case has a molecular weight, a measured mass to charge ratio of 162.112 Daltons, and it elutes in this system at half a minute. That's pretty early, and by most means that would be difficult to identify as a consequence. So first we go into our fraction bank and we see in the analysis that was performed by the MRMS, do we have any matching masses which have been confidently annotated in terms of having a chemical formula assigned to them? Then we go ahead and look for the same nominal mass, and we're able to, in this case, using MetaboScape, look for in those rough fractions a mass which in this case has been already annotated as L-carnitine. So this becomes a suggested annotation derived from the MRMS of a biomarker derived from an LC-MS routine screening assay. So if we actually look at the fractionated component here and we go to NMR to try and look at it itself against the suggested assignment, which in this case is carnitine, we can look at our database of reference materials. We can compare the carnitine profile to the profile of, in this case, just the reversed phase fraction here, so the one eluting very, very early. And we can see that's very complicated. That's fair enough. This is inconclusive. So we look to further our 2D fractionations. In this case, we have applied a Hillock fractionation to all of the original reversed phase fractions from the Biofluid Mapping procedure. And if we look at the NMR profile from one of these more highly purified fractions, we can actually do a better job of comparing directly to the banked reference standard and associated profile. And as such, without doing any actual bespoke experimentation for the example, we're able to pretty confidently say we do have a formula from MRMS, we do have a spectrum match from NMR, and so we're very confident in our de novo assignment here. We're further interested in automating this, okay, because still that's a bit manual. When we have a query, we have to go into all the data and assess it manually. But we believe this can be automated, and in doing so, it kind of looks like this. What we do is we look at the elution profile of our unknown molecule across multiple fractions. So let's say we've oversampled the fractionation, and so our target chemical, carnitine, as it were, would elute a little bit in fraction 43, a little bit more in 44, most in 45, a little bit less in 46, and so on. So we can actually take that pattern, we can integrate all those signals, and we can do a statistical correlation to what's seen in NMR. This uses a technique which was developed within our group back in the early, mid to early 2000s, which was called statistical heterospectroscopy. And the idea there was to actually correlate signals between NMR and MS. And I think it's been a push to do this in very complex biofluids, but in these more simplified pre-fractionated systems, it actually works really, really well, often revealing exactly what we need to know. So we do this statistical purification in order to link the different platforms, which have very different data structures, and that in that way we're able to translate between them to get more confident IDs out. Okay, so putting it all together, when we have an unknown from our UHPLC-MS platform, just by looking at the mass-to-charge ratio, we can often translate into MRMS, pulling out what we know from that technology, which would be molecular formula. Using statistical heterospectroscopy, or SHI, we can translate from either MS platform into NMR. And then, of course, once you're in 1D NMR, it's very easy to translate into 2D NMR and gain structural information from that. So what we hope to do in the future is use these statistical techniques to couple all of these different data sets, which have already been built up for biofluids, to automatically pull all of the information together for more extensive characterization of these biofluids. Okay, so we've talked about spectral deconvolution of quantitative profiles, accelerating de novo structural elucidation by purification and implementation of MRMS and NMR, as well as sort of an extension of that, which is biofluid mapping. So as we move towards having this platform for metabolic measurement, the next step becomes how harmonious is this? How far can we deploy these methods across different technologies, right? So we've worked with Bruker in this space to determine interoperability between their IMPACT-2 ToF system and the ones that we routinely use within the Phenome Centre. And just going very briefly through it, we look at specific spectra as generated within the Phenome Centre, and importantly, as entered within the Phenome Centre databases for assignment. And we then ran the same standards on the IMPACT-2 system with the help of the good folks at Bruker. And we found that, in fact, there's a bit of flexibility here in terms of recreating those profiles. So using the IMPACT-2 system, we find that we have a bit more sensitivity, and that when we dial up fragmentation energy, in this case adding 8 volts to the ion energy, which occurs before the quad, and about 10 volts to the collision cell itself, we're able to faithfully recreate the same spectra that we routinely obtain on our instruments within the Phenome Centre. And what this means, and it's very, very important, we're able then to utilize our databases to help annotate data being run by our standard methodology, regardless of the type of instrumentation, or at least on the Bruker Impact system, as well as the kit that we run. So this is an important thing if we want to share the knowledge that we have at hand, and we want to enable collaborators and other labs to leverage our databases and those investments. We have to make sure that the technologies are interoperable. And our work to date last year with Bruker has demonstrated that to our satisfaction. Interestingly, the IMPACT-2 system surprised us with its reliability and robustness. We were very pleased with that. And just a brief summary of the data quality coming off of our very first test. This isn't a selected test. This is the first one that we did in conjunction with Bruker on the system. Shows that the median relative standard deviation value for all features extracted using the MetaboScape software from a small test set of samples, the median RSD was below 10%, which is very, very good. This is, of course, in the raw data without any correction applied, without any statistical correction or quality control sample selection applied. And so we're, like I said, we're pretty surprised by this right out of the box without much configuration or tweaking. The performance is very good, in addition to the interoperability. So we think this is a nice platform going forward for routine phenotyping measurements. I'll just draw your attention to the study design on the right there, because it's important in a subsequent slide. We for these types of comparisons, we like to establish a relative comparison amongst, in this case, 3 distinct urine samples, which have been mixed pairwise to make mixtures that we know should represent half of one primary sample, for example, half of urine 1 and half of urine 2. Okay, that design will come up in another slide here. But looking at the precision of peak area, mass accuracy, and the isotopic pattern as measured by MetaboScape's M sigma value are all very good, despite the fact that it was a greater than 14-day run and that we're looking at over 1,000 injections here from a cold start. Okay, so how to further accelerate this? This is all well and good, but what if we want to tackle very, very large studies? We're going to need an even faster solution. So we've been working with Bruker on their CyMax MRMS system in order to see how we can adapt our protocols and our sample preparation procedures for application on this type of system, and we've been looking at what kind of data we can get out of this system and whether or not we think it's satisfactory for different phenotyping applications. The long and short of it is that it's actually quite impressive in terms of its throughput, as well as its ability to automatically annotate chemical formula and match spectra obtained using the fine isotopic structure to databases. This becomes extremely powerful, returning very definitive, well-characterized results without much database building. Okay, so we're not, in this case, talking about building large LC databases. We're just relying on the information at hand, which is largely thanks to the extreme resolution of the CyMax MRMS. So I believe there's a presentation after this on MetaboScape, so I won't talk about that in any detail, but I will return to that original study design, which we've now run on the CyMax instrument. And you can see here the fidelity with which the sample metabolome is captured in this principal components analysis here, which faithfully recreates the original study design, where the mixed sample A-B does sit between samples A and B, and so on, right? So we use this as a way of establishing sort of a ground truth on whether or not this system is faithfully representing the chemical content. And I think most impressive is the fact that of the, looking at the bottom table, of the 6,539 features measured by this system in both positive and negative electrospray mode, 4,834 of them were assigned a formula using the Smart Formula Generator, and 488 of those actually have been linked to HMDB list for what would be expected in urine. So this is actually a huge level of characterization coming right out of the box without any database building initiatives, which can be lengthy and expensive. So we're quite impressed with this technology. We think it has a place in future, perhaps biobanking initiatives, large epi study initiatives. So just very quickly want to thank the good folks at Bruker. We've really enjoyed our collaborative work with them over the last year. And that's a few of the more critical members of our team on the right who are specifically have contributed to the LC separations I've shown, the ToF and the MRMS data generation, as well as the NMR. If you want to learn a bit more about molecular phenotyping, we have an online e-learning course, which I'll direct you to here. It's got good reviews. It's open to anyone from an academic institution or medical institution, so please check that out. And just want to acknowledge our wonderful team there and our director, Dr. Zoltan Takach. Okay.
Thank you, Dr. Lewis. Excellent data, excellent presentation. I'll now introduce our next and final speaker, Florian Zubeil. Florian joined Bruker Daltonics in Bremen in early 2018 after conducting his postdoc at the Sanofi Fraunhofer Natural Product Center in Frankfurt, Germany, where he oversaw the MS-based Dereplication platform. He is a natural product chemist by training, interested in the MS-based identification and analysis of small molecules. At Bruker, he is part of the Metabolomics applications team for ToF and ToF Instruments. I'll now turn the presentation over to Florian.
Thank you, Nancy, for the nice introduction, and also welcome from my side from Bremen to everyone joining online. I would like to give you a short introduction about our untargeted metabolomics software, which is MetaboScape, currently at version 4.0. And to give you an idea about what the software is doing, Louis already told that depending on the application that you have, you need different instrumentation to address these questions. And this is why MetaboScape does not only support one instrumental platform, but actually 3 different Bruker-based MS platforms. And they all come with a specific peak picker, taking the strengths of the solutions into account. So the first algorithm would be the T-Rex 2D feature finder for flow injection MRMS analysis, which is for very high throughput phenomics solutions, so you can get up to 200 samples per day. The next one would be the T-Rex 3D algorithm for LC-QTOF data, which also provides quite a high throughput. And as you already saw, we can easily process more than 1,000 LC-MS runs using this solution. And if you want to dig really deep into your samples and also separate isomers and use collision cross-sections, we also have a T-Rex 4D algorithm supporting the TIMS-ToF technology, so the trapped ion mobility, where we can separate isomers and also can calculate CCS values. And the important thing is that in the software, although the peak picking algorithms are different, the use of the software is the same. So as soon as the information is extracted out of the data, they are all seamless integrated into the software. What's also nice about the software is whenever you identify a compound, you can easily see how good the annotation is. So you will always see the small annotation quality score you can see on the lower left side, which will immediately give you an idea on how good the precursor mass accuracy, the retention time, the isotopic pattern, the fragment spectra, and also CCS value matches. And here you can see a screenshot of the software. And the most important part really is the table in the middle, I would say. It's the so-called bucket table. And ideally, every row contains one compound in your samples. And then you can see the intensity of this compound through all of your samples. And this bucket table is built up by the different T-Rex algorithms I just showed you. And then when you have this bucket table, you can easily do statistical analysis. As you can see on the bottom, there's a PCA open. And you can also identify compounds from this point on. And what's quite nice about this software is that it's really interactive. So you can easily see how annotated compounds behave in statistics. And you can also easily follow up to identification if you have some unidentified compounds which are regulated, and that you see that in the statistics. So the steps in the workflow in MetaboScape can be separated in 5 different steps. And the first step, of course, would be to acquire data at all on any of the 3 different analytical platforms. And then what you can see here, what's very convenient is that every other step after acquisition of the data is completely handled inside of MetaboScape. So there's no need to switch the software if you go to statistical analysis or annotation. But as you saw on the screenshot, it's all handled in one software. So it's a really easy to use software to work with. And as soon as the data has been acquired, the next step would be to do the peak picking with the different T-Rex algorithms. And this will do some more advanced algorithms I will show you on the next slide. So it will also perform deconvolution of the data, retention time alignment, and recursive feature extraction to extract all the information from your raw data and to build up this bucket table. And as soon as we have this table, no matter which analytical platform we choose, we can go further on to identify our compounds that we detected, and also to perform statistics to identify regulated compounds. What's also supported by MetaboScape is a visualization of biochemical pathways. So if you're looking into a specific biological pathway, you can import this to MetaboScape, and then you can immediately see if the compounds included in this pathway are regulated or not, and also if they are down or up-regulated. So if you look at step 2, the peak picking algorithm, and here's an example of the T-Rex 3D algorithm for LC-QTOF data. The algorithm will handle quite a bit of data processing on its own. So it will do an automatic data calibration, and this is true for mass position calibration, but also for mobility calibration if you are looking into timsToF data. What the software also does is it will not only pick features, but it will combine these into buckets. So it will automatically detect isotopes, different charge states, adducts, and also neutral losses of compounds. And we put all these features into one bucket. So on the right, you can see there's one example bucket spectrum where you can see that neutral losses and also adducts as well as isotopes have been collected in one single bucket. And this really greatly reduces the data complexity, because otherwise you would end up with a lot of redundant information, because all the signals would belong to the same compound. But at the end, you do not want to look at single signals, but you want to look at compounds in your sample. So what the software will also do is a retention time alignment. So if there are some compounds included in your sample which do not behave very well in chromatography, MetaboScape can handle this by applying a retention time alignment. And it will also apply a recursive feature extraction. And I will show you this on the next slide. Actually, the recursive feature extraction is a really important feature if you want to do statistics on your data. And to explain what this algorithm is doing, let's have a look at these 4 samples. So you have sample 1, 2, 3, and 4 below each other. And then we are looking at 2 different features, so feature 1 and feature 2. And during peak picking in every metabolomic software, you need to specify a certain threshold to distinguish noise from real signals. And we can see that, and this threshold is depicted by this dotted line you see here. And we see that feature 1 is detected in all 4 samples consistently, and the respective intensities are then reported in the bucket table. But for feature number 2, you can see actually a peak is present in all 3 samples, but due to regulation in sample number 3, the peak is just below the threshold. So in the first step, the software would report a 0 in your bucket table. Of course, this would not be good for statistics, because this would assume you would have total regulations, although there still is a signal present. So what this recursive feature extraction is doing is that you can specify a minimum number of samples where a feature has to be found. For example, this could be 3 samples, and then the software will realize, okay, in the 4th sample, I did not find this peak. And then the software will go back to the raw data, lower the threshold, and perform the peak picking again in this specific region with more relaxed parameters. So you will then be able to still get a peak intensity also of regulated peaks which are regulated below the usual threshold. It's important to know that for total regulation, you would still be notified, because you would then still be see a 0 if there would be no peak at all. So for timsToF data, this is also completely integrated in MetaboScape by the T-Rex 4D algorithm. And you can easily detect isomers in MetaboScape. You can get cleaner MS-MS spectra by separation in ion mobility of the precursor ions. And you also can see a CCS value, so a collision cross-section value, for all your analytes. And it's important to know that the CCS values are a property of the ions of your compound. So they are independent of any chromatography. So they can be easily used for annotation of your compound as an additional qualifier. You can also see mobilograms of all of your compounds, so you can, for every compound, easily see how it behaves in the mobility dimension and also if there are isomers present or not. So now we have picked all our peaks in all our samples and we have created our bucket table. And when we want to continue to identify our compounds, MetaboScape gives you quite some tools to do so. The first tool would be an analyte list, where you can basically specify a sum formula and a name for a compound and optionally also retention time. And this will allow MetaboScape already to perform an annotation based off the accurate precursor mass and also of the isotopic pattern. And if you supply MetaboScape with a retention time, then also the retention time will be matched. Of course, this is dependent on the chromatography, and if you want to be independent of this, you can also do a spectral library match, where MetaboScape will judge the fragment spectra match and also the precursor mass and also isotopic pattern. So these libraries can be used for annotation of your compounds, and you can get libraries from Bruker, which have been acquired on Bruker instruments, but you also have the ability to import open formats, for example, the MSP format from Mass Bank. So the analyte list and the spectral library would give you confident annotation of your analytes. And then if you do not have an annotation here, you can still go on and calculate the sum formula of these buckets, and these can be done in batch by the smart formula algorithm. And as an unknown workflow, you can then query online databases, for example, ChemSpider and PubChem, for the determined sum formula, and get chemical structures which are already published in databases. And as Matt Lewis already mentioned, we have the in silico fragmentation of MedFrag completely integrated into the software. So usually MedFrag is an online service where you can upload a spectrum, but in MetaboScape, the algorithm is completely integrated into MetaboScape, and there's no data uploaded to any server, and you can inspect the match of the in silico fragmentation offline in MetaboScape. And when you have an identification in your bucket table, as you can see on the bottom here we would have caffeine identified. You can see we have one molecular formula assigned, and you can also see that this annotation you see is performed based on the blue analyte list, the blue AL icon. And you will also see this annotation quality score, which will give you a quick overview on how good this annotation is. So as you can see, the different tools for annotation in MetaboScape give you different annotation confidence. So the best thing would be to annotate with an analyte list and a spectral library, where you can compare 5 different parameters. So you can judge the accurate mass of the precursor, the isotopic pattern, the fragment spectrum match, the retention time, and if you're dealing with timsToF data, you can also compare the CCS value. And then we also support less confident annotations by the tools I just explained to you. So all these tools take different strengths of the different instruments into account. And this annotation quality score is very useful if you want to have a quick overview on how good your annotation is. So you will see that for every qualifier, you have one entry in here, and you can have 2 green bars or for a perfect match, or 1 gray bar for a medium match, or even no bar if this information is not present in your data because, for example, CCS value is only available on ion mobility instruments, of course. And what you consider a good or a medium match or no match at all can always be defined by yourself. So whenever you do an annotation, you can select by yourself which mass accuracy you trust, and also which isotopic pattern you trust. So I also want to present the dedicated Accelerate workflow that also Matt already talked about. And this is built on the Cymex MRMS platform. And as you already heard, this platform can provide a very high sample throughput. So you can easily measure over 200 samples per day. And this is done by skipping chromatography completely and just recording one spectrum per sample in this very high resolution MRMS instrument. So in this spectra, we will get a resolution of more than 1 million, which enables isotopic fine structure. And also we have a very good mass accuracy of below 0.2 ppm. So the high resolution combined with a high mass accuracy really allows just to record one spectrum per sample, which can be then easily used for sum formula determination and also for profiling of large sample cohorts. So to sum up, MetaboScape is a really intuitive and comprehensive software for untargeted metabolomics workflows. You saw we have different algorithms for importing data into the software based on the different hardware platforms. We have different tools for performing annotations. And also the statistics is all integrated into the software. What can also be done in the software is the merging of positive and negative ionization data. So when you have one batch of positive ionization data and one batch of negative ionization data, you can combine the results into one single bucket table and work with a bucket table containing both ionizations. And with this, I would like to thank you for your attention, and I would like to hand over to Nancy again. And if you have any questions, then please feel free to ask them.
Yes, thank you, Florian. Impressive software. Yes, we're going to take time now to answer any questions that you may have. As a reminder, please use the Q&A box. And we will start. A couple came in that I think might be targeted to you, Florian. The first question, you presented the recursive feature extraction workflow. Is there a way to easily identify recursively picked signals?
Yes, this is very transparent in the software. So you will see the intensities in your bucket table, and every signal that has been picked recursively is in italics, and it's also shadowed. So it's easily seen that this one has been picked recursively.
Okay, I think this one might be for you as well. Is it possible to use unpublished chemical structures like known in-house analytes for the in silico fragmentation, or do you rely on online databases?
Yes, that's a very good question. So we use our compound crawler tool, which can access online databases. But you have also the ability, and this is very important, for example, for pharmaceutical companies, or if you cannot share information to the internet, you can also import chemical structures as SDF files and can then use these structures for unknown identification workflows. So if you have already a library of chemical structures, this can be easily integrated into the compound crawler software.
Okay, thank you. Another one, I think, related to MetaboScape here, you described the importance of isotopic fine structure for unambiguous formula assignment. Can the MetaboScape software you use handle this information, and if yes, in which way?
Yes, the isotopic fine structure is also automatically handled by the T-Rex 2D algorithm. And what we at Bruker always do is we calculate a so-called millisigma value for the quality of the isotopic pattern match. So we always compare a theoretical isotopic pattern with our measured isotopic pattern. And this can be done both with non-isotopic fine structure patterns, but also if we have isotopic fine structure available. And this is then used to rank the correct sum formula according to the best isotopic pattern match. So this is automatically handled by the T-Rex algorithm and the smart formula software.
Great. This one's coming in for Dr. Lewis. You mentioned that the identification of unknown metabolites is one important bottleneck in phenomics research. Can you kindly describe in which way the extreme resolution of the MRMS system can support this task?
Well, that's really coming down to its elucidation of the isotopic fine structure. That's what enables us to specifically build the definitive chemical formula in a way that really even normal routine application high-resolution instruments can't do. Yeah. So there's really no interpretation involved. It's basically counting signals at specific isotopic positions, and it's unbeatable for that purpose.
Great. I think this one was covered in your presentation, but a question for you as well. For processing the LC-QTOF data from the Bruker impact system, you said that the data from greater than 1,000 injections were processed. Did you use maybe MetaboScape for this, and how long did the processing take?
Yeah, so that's a good question. It took us about 2 weeks to acquire the data, but the processing was done on I think on a standard PC. It took about 15 hours, right? So that's basically an overnight process, which is pretty good for that size of data.
Okay, well, I think we're out of time right now. I want to thank everyone for joining us today. If you would like to view this webinar or any recordings of past webinars, you can visit us at www.bruker-webinars.com. We certainly appreciate your time today, and have a great rest of the day. Thank you.
Thank you.